So let's get rolling. All right, so when we're talking about evolution and natural selection, there's three main things that need to be present for natural selection to occur. The first of those is going to be variation. And of course, you have to have variation to have selection, which we'll see when we look at question number 3. You also have to have limited resources or a large population, depending on how you want to look at that, because the limited resources and the large number of organisms, that's going to lead to competition for those resources. And then because there is variation, the organisms with the better variations for getting those resources will then be the ones that reproduce at a higher level. And therefore, they are going to pass on those traits. And therefore, that's going to select those traits for that particular environment and for those conditions. So when we talk about, and so it's not an all or nothing kind of thing. You know, you will still have this competition, and some organisms with the less adapted conditions, some of those still will continue to reproduce. But what we should see is the ones with the better traits reproduce more. And so that brings us to the idea of absolute fitness versus relative fitness. And when we talk about this, remembering that fitness is the reproduction of viable offspring. So when we talk about absolute fitness, that's simply the number of offspring. So in this case, we see 4 offspring. And down here, we see that this is the population average, which is 3. And so relative fitness is going to be a comparison with the average. So where this one has produced 4 offspring, and if the average is 3, then this individual right here, elephant E, its relative fitness is 1.33, which means this one is slightly more successful than the average elephant. So if you are an average elephant and you had 3 offspring, you would have an absolute fitness of 3 and a relative fitness of 1 because you equal the average. And so then you can compare and see who is more fit and potentially see why one organism is more fit than the other. Sometimes the fitness level has nothing to do with the traits, because sometimes natural selection happens due to random chance. But this is why we need variation. If I say, pick your favorite color, and then over here I say, pick your favorite color. You can do that over here. You can't do that over here. Because here there is no option. So if you only have one thing in terms of degrees of freedom, how many would that be? That's 0 freedom. This is the only thing you get. But over here, you would count and subtract 1 for your degrees of freedom. All right, we can also have maladaptive evolution. And this is simply that sometimes traits aren't good. So if you are a gray-furred cat living in a dense forest, that dark gray is going to help you blend in with shadows. You're going to fit in better, help you capture prey. But if this dark gray fur was in a sparse desert, that's going to stick out against that lighter background and be bad. Or again, a polar bear in a tropical rainforest, not going to work so well. Or, you know, this right here outside, not good. Sunscreen, hat, necessary, maladaptive, great for swimming. All right, so then natural selection versus artificial selection. Natural selection, your selective force is going to be the environment. meaning the climate, the weather, the rainfall, the temperature, also the number of predators, how much food, how much water, how much space. All of those environmental factors are going to be the selective pressure, whereas artificial selection, it's going to be humans picking the traits and then controlling the breeding. And so that's how we go from wild mustard to all of these different plants, or from wolves to all the different breeds of dogs. Convergent evolution, then, this is where you have similar phenotypes. Even though they are not closely related. And why do they have the similar phenotype if they're not closely related? Similar environment, right? So this is why morphological choices for things like cladograms are not as accurate. Because if you are looking at physical characteristics, they may have arisen due to convergent evolution, which could make you think that they are more closely related than they actually are. So that's why molecular data, DNA and proteins is going to give you better information for cladograms. Different things that are going to affect natural selection. So genetic drift, both of these things. This is random chance. That is going to eliminate certain alleles. So here, some bugs get stepped on and killed just because, and therefore their alleles are not going to get passed on. Or potentially, with some plants, wind pollination, randomly, which genes get to which flowers, or from which pine tree to the other pine tree, or what grass to the other grass, random, because sometimes those genes just aren't going to make it. That can also occur with fertilization with sperm and animals. So the male is making lots of sperm. So which trait actually gets to fertilize the egg, that's going to be random. So sometimes certain traits can be lost. Then we have bottlenecks. So you've got all of these specific traits, but then you have some event that limits the population. And this is usually due to a natural disaster. So a volcano erupts, a wildfire happens, a large portion of the population dies. And so this is going to reduce your genetic variation. And the same thing is going to happen down here with the founder effect. Where a small group of the population gets separated, and then that is the base for your new population. So you're going to have a smaller amount of genetic variation to start. So this is what we saw in the Galapagos Islands with the finches, and then they do adaptive radiation spread out and separate into different species. All of these reduce genetic variation. So is that good or bad for the population? It's bad. So natural selection can also reduce genetic variation if you have very, very selective pressures, and it eliminates lots of traits quickly. That can also reduce it. So bottleneck again, reducing the population down quickly. Founder effect. So this is showing something that is interesting of how tropical diseases can go to non-tropical places. Hooray for airplanes. So you can have someone visiting Africa, and then the mosquitoes get on the planes with the people. And then they can travel to America. So that small group of mosquitoes, then that's the base for the new population in America. And then maybe this population starts growing and we're like, these are bad. So we use a bunch of insecticides and we reduce that down, and maybe these are the resistant mosquitoes, and then that grows into a new population. And then these are just mutations that are starting to show up. And then you get, and maybe, more selection. So all of that is a combination of founder effect and then bottlenecks. All right, introducing new genes can happen when populations mix. So when you have migration, you can have new alleles with immigration. So these deer are coming in. These are the same species, but they just have slightly different coat colors. So these two new deer are introducing new alleles into this population. You can also lose alleles through migration. So if a subgroup of the population has a particular allele and they leave, that could reduce genetic diversity. So this could go either way, increasing or decreasing genetic diversity, depending if you are getting new alleles or if you are losing some alleles. Mutation is also going to introduce new alleles. Are mutations generally good or bad? They're generally bad. If they're not bad, then they're generally just neutral, such as a silent mutation. But occasionally you get a new mutation coming in. So again, this is not a photoshopped cardinal. There are now yellow cardinals. And if the female cardinals suddenly decide, we like that better than the red, then those traits are going to start to show up more and more and more. All right, so then this takes us to looking at population and allelic frequencies. And we are looking at Hardy-Weinberg. So if a population is in Hardy-Weinberg equilibrium means balance. And in this case, that just means no change in allele frequency. And if there's no change in allele frequency from generation to generation to generation, what does that mean in terms of natural selection and evolution?
They're not being selected.
Right, there's no evolution. So this is actually very rare. So conditions that we need for Hardy-Weinberg equilibrium, there's no selective pressure, which means like this trait is fine and this trait is fine. And that's usually when there is no competition for resources. There's also going to have no mutations, so we're not getting new alleles coming in. Typically a very large population, because the larger the population, the more each allele is represented. And so it is less likely that those alleles will be lost. If you have a very small population, and maybe you only have two individuals that have a particular allele, those two individuals die, that allele is gone. That is less likely to happen if you have a very large population. Also random mating, so there's no selection for a particular trait. So the females don't care, the males don't care. So you'll see that again with a lot of wind pollinated plants, with the animals where sperm and egg are just kind of released into the water and then they happen to run into each other. And then no migration, so you're not going to lose alleles this way. All of these things, when you put all of this together, is that going to be common in nature? No. So the Hardy-Weinberg null hypothesis is going to be that there is no change. So in order to show that there is change, we are going to use the Hardy-Weinberg equation from one generation to the next. And if the allele frequency changes, then we would say we reject the null hypothesis and some type of selection is happening. All right, so there's going to be two types of allele analysis. One where you know all of the genotypes and one where you don't. So when you know all of the genotypes, this is going to be things like incomplete dominance, codominance, where the heterozygote is different from the homozygous dominant. So if you have big R, big R, little r, if these look the same, then we can't tell the frequencies and we'll have to use the other method. But in this case, if we have red flowers, big R, if we have white flowers, WW, if you've got RW, then they're pink. So that's incomplete dominance. And therefore, if we know it, if it's pink, we know it's RW. If it's white, we know it's WW. If it's red, if it's a regular dominance, where that's red and that's white, well, we would know white, but if we saw a red flower, we wouldn't know if it's homozygous dominant, homozygous. So if we do know the difference, then all we're going to do here is tally the number of alleles divided by the total. Very simple. And so how many red alleles would I have in this case? Well, not half of 75. It would be 75 reds and then 100 over here. So right thinking, but it just kind of flipped a little because there's 50 RRs, so that's 100 Rs plus 75. And then how many whites would I have?
125.
I'd have 125. So I'd have 75 whites from the pink. And I would have 50 whites from over there. What is my total number of alleles?
300.
300. So 50 plus 100 is 150 individuals. Each individual has 2 alleles. So that's over 300. So that's 175 divided by 300. And then 125 divided by 300. Did you do the math?
Yeah, I got 0.583 and then 0.417.
All right, so that would be our allelic frequency. If I said, what is the phenotypic frequency? What would I do there?
The number of like for red would be like 50.
Right, so all of this is allele frequency. Phenotypic frequency would simply be 50 over 150 for the red. 75 over 150 for the pink, and 25 over 150 for the white. So you have to look at what the question is asking. Is it asking about the alleles? Is it asking about the phenotype? Now, if we went through the summer, through the fall, and then in the next year, we did this again, and if it was 0.583 and 0.417, again, then we would be at Hardy-Weinberg equilibrium, meaning no evolution. But if it was different, then there was some kind of evolution happening. All right, if the genotypes are not known, then this is where we are going to have to use p squared plus 2pq plus q squared equals 1, and also p plus q equals 1. And if you still cannot remember those equations, guess what? They're on the equation sheet. So what does q squared represent?
This is
Homozygous recessive. P squared equals, and then 2PQ is the heterozygous. So then P represents your dominant allele. Q is your recessive allele. And so when we look at this, you've got 75 red, 25 white. Can I tell which red flower is homozygous dominant and heterozygous? No, I cannot. So I have to look at my white flowers. So I have to figure out my frequency of my white flowers, which is going to be 25 over what? Over 100. And so that's going to give me a frequency of 0.25. And this is the frequency of what? Which variable? Q squared. So then, that's the frequency of Q squared. Q equals, you take the square root of that. And so then Q is going to equal 0.5. And if Q is 0.5, then P is also 0.5. So we just use that equation to solve for P. And then I can solve for P squared and 2PQ. So then P squared is going to be 0.25, 0.5 times 0.5. And then 2PQ is going to be 2 times 0.5 times 0.5. And that will be 0.5. So that is going to be your different gene frequencies. And so then if you have a larger field of flowers and they have the same frequency, and I said there's 100,000 flowers, make it easier, if there's 1,000 flowers, how many of them would be heterozygous? Yeah, so then 1,000 times 0.5 would be 500. So, all right, natural selection, 3 main types. So you have stabilizing selection. So this is going to favor the middle ground. So robins typically lay clutches of 4 eggs because that's what they can handle raising. If they put in too many eggs, then they can't feed all of them and they just die off anyway. Directional selection favors one extreme, either the other. And so in the Industrial Revolution, we started burning lots of coal, and in England, it caused all the trees to get darker because of the soot. And so the peppered moth then shifted to have a darker form. So that's directional selection, so it could blend in with the tree trunks better. And then when they started having cleaner air, it shifted back the other direction towards the speckled, which matched the natural bark color. Diversifying selection, the middle ground is unfavorable for some reason. So the extremes are going to be favored. I don't know why, rocky environments, whatever, but just if it's the opposite of Goldilocks, then it is diversifying. All right, small populations have difficulty because of something called the Allee effect. The Allee effect is that populations that are small tend to be very genetically similar. They also have trouble finding each other if they have a small population. So it's very difficult for them to, in terms of population growth, to reach that reproductive momentum to start growing quickly. But more importantly, so with the Florida panther, you had lots of genetic diversity. Then people said, panthers are bad, and we kill a bunch, and then you get your population down to about 30 individuals who are all genetically related. And then you start getting genetic defects because of inbreeding. And so then to help counteract those genetic problems, they have shipped in panthers from Texas to increase the population size for better genetic diversity. The largest parrot. It is the land dwelling flightless parrot called the kakapo. Lives in New Zealand. About 248 of those left on the planet. Because when humans, got to New Zealand, they also brought rats with them on their boats. And then rodents will go and eat the eggs and eat the baby chicks, cause the population to drop. So now their populations, they're very protected, but their populations struggle because of low genetic diversity, et cetera. Okay, fossil evidence. What we see is a change in form. But typically of hard structures. So things like bones are going to fossilize. Things, what doesn't get fossilized?
Tissues.
Soft tissues, what else? Right, but also things like intelligence and behavior, things that are not physical things, all of which can affect evolution. So part of the problem with your fossil evidence is, there could have been changes going on that we don't see. But typically you can see changes in form over time. So you can see how the horse ancestors' foot was different, and then kind of the middle thing became prominent. We can date fossils two ways, either relative dating or absolute dating. Relative dating is simply saying that the higher they are up in the rock strata, the younger they are. So fossil found here is older than fossil that is up there. There's literally a question on that on the AP exam one time, where they gave you fossils and they were like, which one is older? And my students were like, that one. It was like so basic that they were afraid to answer it. Yes.
Can you explain that one more time?
So sedimentation, mud, and then you get the rock layers build up. So then your older rock layers are below, your younger rock layers above. We can give more specific time frame data for our fossils in terms of years and how old they are. This is going to use radiometric dating and half-lives. And these give years. It's not, when we say absolute, all that means is we have a general estimated time. So like 495 million years ago, that's a lot of zeros. So in terms of significant digits, there's a lot of wiggle room. So it just kind of gives a general age. Yes.
Is that just like carbon dating?
So carbon dating is a form of radiometric dating, but when you're talking 495 million years ago, you're using things like uranium isotopes, which have a much longer half-life. So your basic half-life idea, you've got a sample, it breaks down by half, it breaks down by half, it breaks down by half. That time frame has a specific time. And so then if you know how many times it has broken down, you probably, I would be very, very surprised if you had to do a half-life calculation on the AP exam. I've never seen it. All right, homologous structures versus analogous structures. So homologous, same components, different form. And then this is an indicator about what? All of these things, same ancestor. Whereas an analogous structure is going to have the same function, but a different form, because why?
They're not like related.
Right. Different ancestors. So batwing and birdwing, these are homologous, but birdwing and batwing to insect wing is analogous. So usually, there's a question about one or two of those, one or both of those on the exam in the multiple choice. All right, comparative embryology. This is going to show us that what about the rabbit and the human versus compared to the everything else?
More closely related,
Right, why?
because they're on the stage.
So the closer the development, the more likely they are related. And so because the development is based on the genetics, and therefore similar genetics is going to cause similar patterns for a longer period of time. All right, moving on to our molecular evidence for evolution. We are looking at either DNA similarities or protein similarities, and then the more closely they are related. So if you were to look at all of this data, so each letter represents a different amino acid. And so anything in yellow is common to everybody. And so you would look at the differences. And then shockingly, what would probably be humans are most closely related to what? It's actually most closely related to,
Tomato.
well, it's most closely related. Well, we've got a difference here between the human tomato and the tomato and the yeast. And then everything else between them is the same. So which two things are most closely related? Are the I and the V? Oh, and the yeast and the human. All right, so probably, I mean, that'd be a toss-up. So you could have some questioning about that. But if we look over here at this one, a little easier, our cladogram, who is most closely related? Monkey and human. And this is looking at hemoglobin molecules. And so you probably won't get a chart like this. You'll probably get something you've seen before where they have the numbers of the sequences. So if we look at this really fast, filling out our cladogram, which things would be, well, what would be our outlier over here?
Polylepis.
Why is the polylepis going to be the outlier?
It's like farthest related from the E.
So polylepis with the E ferris is 21. Polylepis with that one is 18. With that one is 17. With that one is 20. So it's the thing that is most different from everything else. So that's our outgroup. And then we've got two things that are going to be closely related. Which two things are closely? We've got two groups that are closely related. Then we see that is the E africanus with the E ferris. And then we see that it's the A force, and then F, that one, and that one. So the question is, do these two go here and these two go here, or do we flip them? Which pair is going to go closer to DP?
The A force and G cells.
Why?
They're more closely related to D.
So D polylepis, 18 and 17, D polylepis with ferris and africanus is 20. So the E's are going to go here, and the G and the A will go there. Where these go, does that matter? No. And then those don't matter either. Those can flip. So you just have to look at your numbers, and you have to look at what's closely related. Things that are highly conserved in organisms. So these are things that are used as proteins to look at very often, or the genes that code for these. And the reasons that these are highly conserved, what do histones do?
They rapidly in.
Right, these regulate DNA. Hemoglobin for oxygen, cytochrome C is part of the ETC, and that's for protein synthesis. So these are things that are all extremely important for life to exist. So when we say they are highly conserved, that means what?
They stay the same.
Stay the same. They're going to have few mutations. So these are things that are going to show much stronger relationships in organisms. So if you had a question on the AP test, which would be a better protein or gene to look at, something that is very, very important for everything, or hair color, not necessarily important. All right, humans, hooray. So our impact on other species, we kill them a lot. So we either hunt them to death, which has happened a lot, or we have our development of our cities has caused them to move and migrate. We introduce new and invasive species to areas all the time that can then outcompete other species. We use drugs to help us, but that can cause antibiotic resistance or potentially pesticide resistance. So if we overuse pesticides in agriculture, that can then lead to insects becoming bigger and stronger. Climate change, so rapid change of the climate that gives organisms less time to adapt. Artificial selection, so we are changing those species for our own purposes. That is very, very rapid evolution. And then domestication does that as well. So cows were not as big as they used to be. We have bred them to be bigger. We have bred chickens to make meat fast. So there are some chickens that can't walk because they have so much protein on them. And so we have bred chickens to lay more eggs, all kinds of different things. Let me pause.